Okay, let's get started. It's a pleasure to introduce Yinon, Rudich from Weizmann Institute. Yinon graduated from Weizmann Institute, then spent a few years in Boulder, Colorado, in the US. And since the late 90s, he is in Weizmann Institute. If you want to see a very long CV, you should go to Yinon's website. I won't go into too much details, but he really got enormous amount of prizes and awards, and spent many years in many different roles in Weizmann Institute. In the last 4 years, he is the dean. And yeah, it's a pleasure to have you.
Thank you, Guy. Thank you all for coming. It's a pleasure to be here in this department and to give you an excursion into something that is highly multidisciplinary. I hope it will resonate with you guys. What I'm going to talk about today is basically something we call the atmospheric microbiome. And this is the population of bacteria and fungi and other microorganisms that live in the atmosphere. And specifically, we are going to look into desert dust as a vector that transports microorganisms in the atmosphere. So a little bit of background. So when we are talking about our rotating Earth, what we have is, due to the rotations, the formation of the Hadley cells. And when we have the Hadley cells, we have convective air that goes up around the equator, but then it descends here at around 30 degrees. And when it's descending, it actually goes sort of an adiabatic heating. We get descending air, which is warm and dry. And that leads to the formation of what we call the desert belt. So we have deserts, both on the northern and southern hemispheres. And these large deserts are sources of dust emission. And we have in the oceans and in the other regions, we have where the deposition areas where dust can be deposited and affect all kinds of biogeochemical cycles. Usually people study the mineralogical aspects of the dust. As you can see here in this simulation, you can see the dust which is being transported along this belt. I'll focus mostly on the northern hemisphere. And you can see that the dust is transported over very, very large distances. And there are teleconnections, for instance, from the Baudelaire Depression to the Amazon or to Florida, depends on the season. And you can see that dust also mixes with other gases and other particle types in the atmosphere, leading to modification of the dust. And then the dust is deposited away from the source region, but really after traveling for long distances, and so it can affect the remote places where it is deposited in. So this is another example of dust transported from the Sahara over the Atlantic, and we can see here what's known as a dust layer. We can see it here with a satellite, a light airborne LiDAR. You can see this dust layer that is going, according to the season, either to the Amazon or to Florida. And here is an example where we studied together with Ilan Koren many years ago. We've done some back trajectories and also remote sensing follow-up on dust storms that went from the Baudelaire Depression all the way to the Amazon forest, where we had some samplers and we could see the increase of nutrients that came with the dust in the Amazon. So this is a teleconnection where the Sahara is actually supporting the nutrient supply to the Amazon forest. And a recent paper with a lot of compilation showed that until up to 2000 there was a global increase in dust, in atmospheric dust. Most of it was from Asia and North Africa, most of the increase. And generally the dust has a global radiative cooling effect. Now when we're talking about the Eastern Mediterranean, we are talking about a very interesting crossroad for many different types of transport from desert regions. We have desert regions here in the North Africa, Libya, Algeria region that is transported to Israel, but we also have dust that comes from the south and eastern part, from Saudi Arabia. We have also dust that arrives from the Iraq, Iran, Syria area, an area that is undergoing desertification and increasing actually in dust activities. And we also have a lot of flow from the west, northwest, which is not dusty, we call it more clean air. It goes over the Mediterranean, a lot of stuff is being deposited. So in contrast to these trajectories, we have more clear air. So when we're talking about the dust, I believe that a lot of the properties that dust has is related to what's absorbed onto the dust and not necessarily from the particles themselves. One of the things is that we have what I said before, the atmospheric microbiome, where you have a lot of microbes that live in the atmosphere. You have, of course, bacteria, archaea, fungi, viruses, etc. And some of these can adhere to dust and can be transported over large distances. The problem is that atmospheric microbiome is one of the least explored ecosystem because you don't have usually a high concentration of biological material. So it is a challenge, but there is a lot of new developments in biology that allow us now to adopt them and try to study the atmospheric microbiome. And the questions that people are asking are, what's out there? What kind of microbiome we have? How it changes? What is their potential impact on ecology, on climate, on health? And what influences their behavior and also the dust? Particularly dust, because it increases a lot the concentration, can act as a very important carrier for microorganisms over long distances, and it can connect remote ecosystems if these deposited bacteria, for instance, can participate in various processes. So it's actually a very interesting question because it is related to global climate change, which the Eastern Mediterranean is a hot spot for. We are undergoing tremendous changes, very fast changes of warming, drying, and there are changes in the patterns of transport of dust. And so all of this is related and can have consequences for biogeochemical cycles. So the motivation of this talk is to evaluate how the transport of microorganisms within the atmosphere influences their composition and viability. We want to assess the presence of potential pathogens for people or for ecosystems, and how they are related to the pathogenicity, so the function of disease transfer, and also for antimicrobial resistance, which is a very important problem in public health, both for people but also for agriculture, etc. One of the things is ice nucleation by atmospheric bacteria. We want to understand ice nucleation. If we have time, we will talk about that. And then I'll talk about new approaches to characterization and prediction of dust events by machine learning and AI techniques if we will have time. So basically, how do we study bacteria in dust? Basically, we sample. We can sample on filters or directly into various liquids if we want to look at viability, or if we want to preserve some of the genetic material. We do all kind of analysis, chemical analysis of tracers, metals, etc. Then we extract the DNA. Oh, sorry. I'm going the right direction. Then we extract the DNA or RNA. We can do something which is called PCR. It's a way to quantitatively amplify genetic material. It allows you to know how much there is. But then we can do something which has become more of very common in biology. We can do all kind of sequencing. We can do shallow sequencing or metagenomics, deep sequencing, in order to understand what do we have there. So that is related to actually blast the DNA and then using bioinformatics to build back the genome and try to see how the genome depends on various parameters, as I will show you. One thing that we can do is also sample on a filter, but in a size-resolved way. So we don't have to take the entire bulk. We can try to separate the different sizes and try to understand at which size range the bacteria reside, how that tells us something about their transport, etc. So we do the extraction. There is a protocol. We can do DNA and metagenomics or sequencing or RNA. The reason is the following. DNA is a very stable molecule. RNA is a not so stable molecule. So we had an assumption that by looking at the ratio between RNA and DNA for specific species, we can get some information about the viability, because we say if a bacteria is dead, its RNA is going to be degraded, and if we find a high ratio, then it tells us that potentially these bacteria are alive. This is a proxy for viability. There are other ways to study viability. For instance, people can grow deposited bacteria, but then you are very limited, because not all bacteria grow on the medium that you're using. Some bacteria use antibiotics to fight each other. So if you want to get sort of a broad view, people are looking for proxies for viability. This is the proxy that we have been using. So let us look at the diversity and richness. So how many types of species do we have in the dust? And what you can see here, when you compare both, the yellow is the DNA number, and RNA, and the RNA is the blue. And you can look at the diversity and richness between what we call clean conditions. I remind you, these are the western trajectories, and then the Saharan dust, and then these are the eastern storms, the storms that come from Syria and from Saudi Arabia. And what you can see is that when you look at the diversity of species, so how many types do we have? We see that we have a higher diversity, substantially higher diversity in dust events compared to the regular events. And then we can look at how many species we have, and we again see the same behavior. So the air mass origin, if it's from a dust source or is over the ocean, the Mediterranean, is the most significant factor that affects the community composition. And another parameter are particle size class and the concentration of particles that are below 10 microns, called PM10, are also significant factors. And we see that that the diversity and richness are higher in those trajectories. We can also look at I told you that we're using qPCR, so where we actually can say quantitatively do we have more or less. And what we can see here, that when we look at the air mass, when we look at the air masses, again, we see higher numbers of DNA and RNA in the dusty events compared to the clean events, substantially more. And you can see that in general, we have a lot of RNA compared to DNA. And also, we can try to see in which particle size they reside. And we can see that as we go from the fine to the coarse, there is a higher concentration of the of bacteria. So that indicates to us that the bacteria are not transported by themselves in the fine and small fractions, but rather they are either attached to the dust particles or are being transported as agglomerates in some soil material, because a lot of these bacteria form what we call a crust, and so when you move it to the atmosphere, they are being transported as agglomerates, so mostly in the large size fractions. We can also look at which types are there, and we can see that we have a lot of soil-dwelling bacteria. So we can see in the southwest, in the Sahara, and in the eastern trajectories, we see a lot of soil-dwelling bacteria, which makes a lot of sense. And some of them are actually human and agricultural pathogens. We will look later on these pathogens. When we look at the northwest trajectories, the trajectories that go over the Mediterranean, we actually start to see also bacteria that are more common in aquatic habitats. This is some kind of an indication for exchange between the ocean and the atmosphere. There are also studies, for instance, by Yal Rahav, who looked at the deposition of bacteria from dust into the ocean and see if they can affect the marine environment. So we can start to look at each one of them. We can see their distribution. And say, I don't want to go to the detail. I'll give you just the broad view. And then we can also look at the species that reside in the coarse fraction compared to the fine fraction. We do find species that are also common in the small fractions. And we find that a lot of these species that are in the fine fraction, which are probably transported as individual microorganisms, a lot of them are spore-forming bacteria. And we believe that the fact that they can form spores or that they attach to the large particles, these are mechanisms that allows them to actually protect themselves from the UV radiation and also from the dry conditions of the atmosphere, and potentially help them stay viable for the transport of a few days from the Sahara to their deposition place. So here we looked at the different air masses and looking at the RNA compared to the DNA in these both the different trajectories and different also the different size regions. And and what we find is that when we look at this ratio, we have a lot of species. This is the ratio of RNA to DNA. A lot of the species have high RNA ratio, indicating to us that they are potentially viable. And now we need more studies to try to start to identify or isolate these species from the entire population and grow them and see if they are indeed alive. And as I said, some of these potentially viable species are pathogens or belong to families of pathogens for animals, agriculture, and humans. So in order to answer which pathogens we had to go deeper in the into metagenomics analysis. So here we get only the species, the family level or genera level. Now we will go down into species level in order to understand which pathogens we might have in the dust. So here we sample in a different way. You sample directly into liquid, and that allows us to take it directly to shotgun sequencing. We will blast the DNA much more, and then we use bioinformatics to understand which specific pathogen we might have in the dust. So what we find is something very interesting. So here in this list, the colors code pathogens, and here I'm showing only pathogens, specific pathogens that we identify by the bioinformatics. The green are for plants pathogens, blue for humans, red for animals, purples are for animals and human, and magenta for plants and human. And you can see that we cover all of this. Overall, we found about 120 pathogen species, and we found, I'll show you later, that they are what we call dust signature taxa. So they're always, or they're correlated with bacteria that are always appearing in the dust. Now, if you ask what are they doing, so for Fusarium, for instance, that's actually algae. It's not bacteria. It's a fungus that affects plants. We have Aspergillus fumigatus, which is responsible for a lot of breathing problems that people have. It's actually very strong allergen. You have botrytis, you have pneumonia, and you have other species that affect people's life. And here we looked at the correlation of these species. This is the abundance. We look at a correlation of the species with temperature and with PM, and we can learn what affects their presence. We can see also dependence on temperature. So as the temperature increases, some of them appear more. So we identified here, so we cannot see really high increase in their concentration per se. It's very difficult to quantify because anyhow, you don't have many. But we look at correlations between their abundance and with the species that we call dust signature taxa. So we found out that there are specific bacteria, soil-dwelling bacteria, that are always there when we have dust storms. So that when we have them, we know that these species come from dust. And then we can look at the correlation between the presence of these pathogens with this dust signature taxa. And again, we have a lot of pathogens that are related to respiratory diseases and to weak diseases. These are mostly linked to dust storms. These, for the wheat, are linked to rising temperatures. So what I want to emphasize is that this provides us a tool to start studying when we expect to see some pathogens. Maybe we can predict them. Their concentration is not very high, but you know, if you have a population that is more sensitive, like elderly people or some people with diseases, they should be aware that if you go out during a dust storm, you have a higher chance to being exposed to this kind of pathogens. We also find a significant increase in what we call functional traits. So what type of pathogenicity, what kind of diseases, and we can get this kind of information. And also we can find that there is a significant increase in species that have resistance against clinically important antimicrobial medications, and like beta-lactams, tetracycline, and all of these antibiotics that are being used to heal all kinds of diseases. What we find is that during dust storm there is increase in the atmospheric abundance of bacteria that have antibacterial properties. Now this is a very important global health issue. There's increase in antibiotic resistance, and we're looking into the potential of dust storm to introduce species that are resistant to antimicrobial properties. This is just to show, you know, how do we verify or how do we know that our assignments are correct. So this is just an example, and I just want to give you it just to see, so you can see the complexities. Here we map the different fragments of DNA into the genome of one of these dust common bacteria. And what it shows basically that all these small fragments cover the entire genome of this species. This gives us more confidence that the assignment of the bioinformatics is indeed meaningful. And it's not that we just find a small fraction from the genome and I say, okay, we have that species. So one has to use a lot of checks to make sure that indeed your bioinformatics is meaningful and not just a result of the, I would say, the bioinformatics manipulation. And here we also we map the genes, the gene types all the way to the species level, and we can see which ones are related to the ones that we actually find in the dust. So these are all kind of checks that one has to do with all this data that you get to make sure that it is meaningful. Now, one of the common the bacteria that we can find it in the, or one type of bacteria that we can find in the atmosphere are actually bacteria that have very unique properties that can lead to ice nucleation under what we call high temperatures. As you probably know, when we have clouds, they can supercool to very low temperatures, down to minus 38 degrees. And if the water is very, very clean, then the water doesn't freeze. It is supercooled. So usually one needs a surface, such as dust, to promote ice formation. But it turns out that there are bacteria in the environment that actually can lead to freezing at about minus 5 degrees or minus 10 degrees because they have very special proteins on their surface that can promote ice nucleation. This is something that has been studied for many, many years, since the 70s and 80s. Zev Levin, for instance, was in Tel Aviv University, has identified some of these bacteria also here in Israel. And one of the questions is, how do these proteins promote ice formation? And here I want to show you a study that we conducted on these proteins in bacteria in order to understand how the structure of the protein promotes ice nucleation. So this is a model of this protein. This model was generated by something which is called AlphaFold. AlphaFold is an amazing revolution in biology that allows us, using a Google tool, to study structure of proteins. This is the best tool in the world now to identify structure of proteins. And this is the protein. So we have here a residue. And then we have another residue, small residue. But in between, we have a lot of points that have a structure, a specific structure that we call it the water organizing motif, because it has here polar groups that allow water to organize along the protein in a structure that is very similar to the structure of ice. And it is believed that once you start to organize the water molecules and you reach a critical cluster size, you can spontaneously form ice. So together with a collaborator, Peter Davis from Queen's University in Canada, Thomas Cook from Germany, and Ido Braslavsky from the Faculty of Agriculture, we were set to study how the structure of this protein allows ice nucleation. So again, here we have the N-terminal domain, then you have the water organizing coils. We have here 65, and then the C-terminal domain. And the way we went to study this, what we did a lot of manipulations, genetic manipulations, to the protein, expressed this protein in bacteria, and then studied how these bacteria promote ice nucleation. So one of the things we've done is we wanted to see how the size of this active site affects the ability of the protein, and I remind you, this is a protein that is expressed in live bacteria, to form ice. So what we do, we grow these bacteria, we put them in our instrument that measures ice nucleation. It's instrument which is called Wisdom. It we make very small droplets, we cool them, and we study nucleation. And we did it actually, this experiment was done in the Wisdom nanoliter droplets, very small droplets that can reach a very cold temperature, super cool temperature before they freeze. And also through our collaboration with a different instrument that has larger droplets where the homogeneous ice nucleation temperature is higher. We did a double-blind experiment. We didn't know what are the samples. He didn't know what are the samples, and this is the result. We can see that when we go down from 65 to about 32 coils, right? You remember? So this number of coils. There's hardly any change in the temperature of freezing. But then if you go below, you can see that the protein becomes less effective and it doesn't lead to ice nucleation. So we see that the size or the length of the active site is important for determining the ice nucleation. We could also do mutations where we change some of these, actually two of these amino acids, threonine and tyrosine, which are the ones that connect to the water into the structure that is ice-like. When we move these threonine and tyrosine, we also decrease the temperature where the ice forms. So these two amino acids are really, really crucial for the formation of the ice, and their presence determines that the water can indeed organize around. Another thing that we've done here is that we found that this C-terminal domain, this end of the protein, is also very, very important in the ability of the protein to form ice. Now, this is very interesting because this part of the protein actually it does not have any more of this water organizing motif. It is actually has a lot of arginine. It's a different type of amino acid. It is positively charged, while this is negatively charged. But we found out that when we remove this part, it is there's no ice nucleation or hardly any ice nucleation. I'll show you that. But something very interesting, just to show you, when we look at all the proteins for ice nucleation and we look at the length of the active site, we see that this length varies from about 70 to about 40. I remind you that below 32 we found that it is actually not very effective. And there is a high variability in the kingdom of bacteria of the length of this protein. But when we look at this one, which I remind you is not water binding domain, it is constant along all the species, 7 or 8 coils. So what is the role of this what we call the R coil, or the positive coil? So we did the same type of experiments. We again generated genetically modified proteins where we reduce the size of this R coil while maintaining the length of the entire protein. And what we found here is that when we go and we reduce the size, we go to 12, 10, and 8, it hardly affects the ice nucleation capacity of the protein. But when we further reduce the size to 6, 4, or less, we see that immediately we lose the ability of the protein to form ice. And the question is why? Why do we have this positive domain? We also asked ourselves, what happens if we take this domain and move it to different parts of the protein, of the active site? And again, we see that we go down in the temperature, and once we insert it here inside, then the protein cannot form ice. And so what we believe is happening is the following. Actually, when we look at the cytoplasm or the surface of these bacteria, we see that there are actually proteins that are very long and large. And we think that this positive part allows the proteins to actually arrange in bind basically one to the other. So the positive part binds next to the negative part, and these are a few configurations. And by doing so, they can actually form these fibers. And these fibers actually add another dimension to the surface on which ice can form. So it's not only the length of the protein, we need the whole surface in order to form these two-dimensional critical clusters that once they form, they can spontaneously lead to formation of ice. So our studies suggest that although this coil, the R coil part, which is positively charged but doesn't have the ice binding domains, is important in the overall structure of the protein. These fibers are consistent with the cryo-chem studies that we've done, and we think that this allows to have a surface on which the ice nucleation occurs. Finally, we have a few more minutes, according to what Guy told me, 45 minutes. I'd like to go and describe to you some of our studies by which we try to understand. So if we have all these important features of dust, can we learn something about dust that was not known before using new tools that are becoming more available now, machine learning and AI approaches? And here I want to show you how we use machine learning to study the meteorological conditions that lead to the formation of dust storms in a way which is new. Before, meteorologists and climate people looked at all these meteorological conditions, where is the low pressure, high pressure system, and then tried to understand under which conditions we are more susceptible to having dust storms in Israel. So what we've done here, we said something else. Let's do it in a way that we don't need to have meteorological knowledge. Let's try to use big data that is available in order to study in an unsupervised way, in an automatic way, what are the factors that lead to dust storms. So what we did here, we took PM10 data, PM10 particles less than 10 microns, dust, from 35 ground stations in Israel. We have data from 2003 until now. This is the record. And then we can take ERA 5 reanalysis data for the entire Mediterranean region, or CAMS, which also is reanalysis data. And then you can have maps, basically, of all the parameters: winds, temperature, relative humidity, and different pressures, what is the PV, etc. We can also use sea level pressure, PM10 concentrations. We can use remote sensing. And finally, we also introduced another data set from Shira Raver-Rubin, another colleague in our department, who studies something which is called dry intrusion, which is basically a phenomenon, meteorological phenomenon. She was your student, right? So this is a phenomenon where air descends from very high altitude, close to the tropopause, and in a few days it goes down, down to the surface. It's a synoptic scale phenomenon, and by linking this upper tropospheric dynamics with the surface, Shira believes that these dry intrusions are important and can explain a lot of extreme weather events such as strong surface winds, changes in the boundary layer stability, high impact weather such as fires, heat spells, etc. And one of the things is that it might be related also to dust activity. So just this is an example of some of these dry intrusions. And this is an example of a dry intrusion that was studied. You can see the air going down, the trajectory is going down to North Africa. And you can see this formation of front that is associated with this dry intrusion. So when the air goes down, it leads to higher winds, and that can augment the dust transport. So what we do here in this study, so we took all that data, we developed an unsupervised machine learning approach. So the idea is, again, let the data tell us the story, and basically we've done some, this is just sort of overview of what we've done. We looked at all these huge amounts of maps with all the meteorological parameters. This is a huge amount of data, so we reduced the dimension in order to have manageable data. We've done some unsupervised clustering to different conditions, and then we can start to look at each one of these conditions and see what they mean. So here is basically how we reduce the dimensionality. Basically, we take all these conditions, we separate them into their major components. You can see that this is different types of components with different length scale. We go from a very long sort of length scale all the way to very high or very small length scale. And basically, what we can do it from the day of the event all the way 3 days before, 7 days before, etc. And now, once we've done that, we can take, we had 256 principal components. We can see that by reducing the dimensionality, for instance, here we use 25 principal components. We see that it doesn't represent very well the observations, but when we use 125, we already get the main features, and so by doing so, we can reduce a lot of the data that we are using in order to do the clustering. And then we were able to do this unsupervised clustering. We found 6 major types of storms. Each one of them has a different seasonality. You see, these are the months. This is when you have the activity. Some are in the summer. Some are more in the transition period. We can get the average behavior in terms of how long it takes. So this is the event, and these are a few days after. We can see what is the concentration. You see that they have different behavior. And then we can start to see where is the dust coming from and how it is related to different meteorological conditions. And this is an example for the deep Mediterranean cyclone that we see. We can identify the cyclone moving from North Africa eastwards towards Israel. We can look at various parameters. We found that this one, for instance, occurs once or twice a year. It has a very long features. It's mostly at the late winter, and it leads to very high dust loading also in southern Europe. And we can identify the wind patterns, the temperature patterns, etc. One of the important things that we find is if you look at these purple shadings, these are actually dry intrusions. This phenomenon that Shira has identified, and we can see how the dry intrusion is moving and pushing the dust by augmenting the wind pattern. And we can see that these are actually important features in several of the, not in all of the components, but in some of them, and driving the dust that comes from different areas. Again, here you can see the dry intrusion, and you can see the dust progressing from Africa to Israel. And it is also true for dust that comes from the east, from the Persian Gulf. So I'm not going to go into details, but then we used AI also to use the data and the knowledge that we got here to actually predict dust storms also in an automated way. We get quite good prediction of dust storms that are driven by large-scale circulation using this AI approach. And we have a lot of activity in my group now working on AI, using AI for Earth and planetary purposes. One of the things is that people tell us always, oh, the AI is a black box, you can't learn about from it a lot. So first of all, I showed you about the dry intrusion. Another thing is using something which is called explainable AI, where you try to see which features the network assigns importance for predicting the dust storm at different times. One can look at different time scales and see if it is the vertical wind, if it's the east-west wind, if it's the mean sea level pressure, etc. So that's a whole feature that one can study the network and try to see what are the important features. So I'd like to conclude here about dust and atmospheric microbiome. So we saw that soil-associated bacteria are transported by dust storms, and different sources actually have unique signature. I didn't show you that, but we can see by the signature if the dust comes from the Sahara or from Syria. It has a little bit of different ecological features. Dust storms increase the amount and diversity of airborne bacteria and increase human and ecosystem exposure to bacteria and biological materials. Specific proteins in specific bacteria actually lead to efficient ice nucleation, and we found the active site's length and specific chemical composition. I remind you that changing amino acids can actually have an important effect on the ability to form ice. So these parameters are crucial for effective ice nucleation by the bacteria. And the aerocoil, the positive part, has a unique and important role in forming structures that lead to ice nucleation. And then I showed you new unsupervised method for clustering dust events in the Eastern Mediterranean. Most of the dust events originate from cyclones that are traveling eastwards. We can find that as well. Mostly dry and warm in the lower troposphere, high humidity in the mid-troposphere and above. This is also a common feature. We found that dry intrusions can enhance dust events in the area. And the thing is that some of the events are mixed, and then it's much more difficult to identify the specific events. So in our study, we actually looked at events that are well separated. And also another thing is that we did the AI method for predicting dust events. Again, this is only predicting dust events that are determined by large-scale meteorological parameters. If it's a local-scale event, some dust, local dust, this approach cannot account and predict these dust storms. So here, these are the people who actually did the work. Burak did the work on the microorganisms together with Daniela. Dori and Ron are working on the machine learning and AI study, and Ma is working on the ice nucleation of the protein, and I'd like to thank you for your attention. Thank you very much.
Yes.
Yes, and we have any questions. So how long can these bacteria live in the atmosphere, and can they get above the boundary? Like how high in the atmosphere can they actually get? Can they shoot like ice clouds in the upper troposphere? Or, I mean, is the lifetime of these bacteria long enough that they can get in the upper troposphere?
The dust events?
Yeah, or just the bacteria in general. Like, I mean thinking shooting ice clouds, like, I mean,
Usually they are not lifted to very high altitude, so I think the highest ones are the ones that cross the Atlantic. So they have to travel at high altitude above the raining clouds. So they reach about 5 to 6 kilometers. So they don't reach the upper troposphere or the tropopause. It's not like a volcano or fires where you have so much convective activity that breaks them. They're mostly driven by ground level winds, and so usually they're not transported at very high altitudes.
So they are not affected by the UV rays and
the bacteria?
Yeah.
So maybe I omitted it. About 15% remain viable. Most of them die. But we believe that the mechanism of spore formation and also attachment to larger aggregates, soil aggregates, can keep them alive, at least some of them. So the lifetime of a dust storm that comes from the Sahara to Israel is about 5 days.
The bacteria.
Yeah.
So the bacteria can be alive for 5 to 10 days.
So about 15% of the bacteria, when they reach Israel from the Sahara, so 5 days, are alive. We think they are alive. We actually